Buriban, and we will continue our discussions of the clinical trial. So the lecture two, it will be continuous of what we mentioned in the last lecture. So we mentioned that the trial design, it composed of first allocation or randomization. Is that trial randomized? And we will discuss what is a randomization and all these types. And the other thing is if it's intervention model and control, like the arm type and the number of the arms. The other thing is a masking and blinding, the type of the masking, like if it's open label, single blind, double dummy, and the outcome measures, or we can say it's endpoints, if it's a primary, secondary, exploratory endpoints, and the study population, who is enrolled. So these questions will be answered today. So randomization. The randomization, actually, we have three types of the randomization. We have a simple randomization, we have a block randomization, and we have a stratified randomization. The simple randomization is there is no categories, it just with no conditions, no categories, just simple without any thinking. So it's like AP, 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 okay? And block randomization, same number in both groups. Like I want a group A and a group B, and each group has the same number of people or volunteers or whatever, but it's despite of their characters, of their age, of their some special characters, I say just the same number. But the stratified randomization, it will be subdivided according to the gender, to the age, to the biomarker level, to the study site, to the period treatment. So it will be subdivided according to the characters. So again, simple randomization like this. There is no categories, there is no characters, just simply. But the block randomization, that we subdivided these people into blocks that they have the same number as we can see here, the same number of the people. Randomization errors might happen longitudinal or between the study centers. So the randomization errors can be happen, and there will be stratified randomizations. Also, the randomizations error might happen between the gender, age, or pre-existing conditions, but they are subdivided according to the maybe gender, age, or pre-existing conditions, but also the errors can be happen. So randomization, so the first point, that is subdivided into simple, block, and the stratified randomization. Now we have something which is called randomization method. There is a randomization method that we can hear it. It's like IXRS and IVRS and IWRS. So we wanted to know what these are. IXRS. I, that's mean interactive. RS, that's mean response system. So they are all shared with the I. So I interactive, interactive, interactive. RS response system, response system, response system. Now we talk about the X. X, it means voice and web. And when it's V, that's mean voice. And when it's W, that's mean web. So we can talk about the randomization methods because we are talking about first the randomization types. We have simple, block, stratified, and now we're talking about the randomization methods. So we have IX. So IV, that's mean interactive voice response system, interactive web response system. But when they use both of them, voice and web, we say X. So IXRS, IVRS, IWRS, these are the randomization methods. So IXRS, interactive voice and web response system, IVRS, interactive voice response system, IWRS, interactive web response system. Okay, so we want to discuss how that is can be going on. What are these methods can be used? So this is the investigational site. will be the investigator, which could be the physicians or dentists or whatever. And here is the drug distribution depot, and we wanted to know the connection between them. So consignment details, it will be to go to the clinical phone inventory database. That's what I mean here. Consignment request. So from them, it will be happen as consignment request, and they will be deal or they will be response by consignment details. And here, that is the important things. The investigational side, they give the notification arrival or damaged packs. They will send to the clinical food inventory database. I mean, all these things, they will be sent that there is, okay, the packets are arrived or there is a will be damaged in the packets like this. They call dispensing call, and they send randomization and stratification back number. These are, maybe we can say, the activities of this method or these methods that we use it. It's IXRS, IVRS, IWRS. These are randomization methods. Register a new patient. There is another topic that we talking about, the register of a new patient. Activities related to the existing participants, confirm receipt of the product, emergency unapplied confirmation, and here it said only accessible by investigator or global unapplied, or we can say medical methods. So here we're talking about um, when we register, maybe these are the actions or the activities that we register a new patient or a new participant. And sometimes we are responsibilities for the activities that is related to the existing participants, like register this participant as a screen failure, randomize this participant into the study, register scheduled resupply visit to this participants, register unscheduled resupply visit for this participant, replace lost or damaged products for this participant, register the end of the investigation product of this participant, register the end of the study for this participant, confirm the receipt of the product, and emergency on blinding information, like emergency on blinding information only accessible by the investigator or global unblinding or medical safety. I mean here all the functions we can say of these things, like IXR, interactive voice and web response system, and interactive voice response system, and interactive web response system. They are responsible for register of the new patient and responsible also for the activities that is related to the existing participants, and they confirm the receipt of the product. But the only thing that is important, like emergency on applied information, like this information should be blinded. That's mean maybe the only investigator know it or someone that is know it, but the participant they do not know it. But it happened some problem during this clinical trial, and this is emergency. And now should the participant know this information? Unblinding of this information, or I have now to tell the patients or the participants about this information. This is the responsibility of two things, of two persons, the investigator or the global unblinding, or we can say medical safety team. If there is a pleading, if it's there is a team, so the global unblinding or medical safety. So again, the response system, whatever is it, is web or voice or something, these are the responsibility of register a new participant, and also activities that is related to the existing participant, confirm the recipient of the product, but emergency unblinding, it's a responsibility of the investigator or a global unblinding, or we can say medical safety team. Now we talk about the control arms, like what we do to the controller group, okay? So something is called the placebo, and we know that the placebo is not active on the treatment, but you can say that the participant think that is a treatment, but it's not, and it has no effect on the body. Standard of care. There is something is called the standard of care, existing a drug for the same condition. Maybe we can give the patients like existing a drug and he think that is a treatment or a new a treatment, and sham a procedure, fake intervention, or fake surgery. All they are, we can say, like that participant think it's a new treatment, it's a new intervention, it's a new something, but it's not, and it has no effect on the patient. Just we wanted to know what is the results of that and how the patient think is it, okay, and how it's affect its condition. I was just making a compare between the control and the case. These are what we can say it's a control arms when you just asking. If you think that the only thing that we give the control is just a placebo, you are wrong. There are three things that we can. Something is called the placebo, something is called the standard of the care, and something is called the sham procedure. And we wanted to know each one what that's mean. Placebo, like we give a drug, whatever is tablet or whatever is a liquid, and we give to the patient. The participant think that is a new treatment, but it's not. It has no effect on our body, and that's what we call it is a placebo. And then the something which is called the standard of the care. What is the standard of the care that we give the existing the already existing drug to this participant, but maybe we can change the label or something, and the participant think that is a new treatment. So that exactly what I mean exactly by the standard of the care. And there is something is called the sham procedure when I make a surgery or a procedure and it's a new, the participant think it's a new, but I just make a fake surgery, and that's what we call the sham procedure. So again, what are the control and all these things that they give to the they are given to the controller group. So the control arms is a placebo, the standard of the care, and also the sham procedure. Now we talk about the placebo. Demonstration of efficacy, measure absolute efficacy and safety, minimization of expectation, ethics, especially if it's a cancer patients, no comparative information. Okay, so we want to know what is we can say what is the goals of the placebo or how it's important the placebo. The first thing is that demonstration of the efficacy, and efficacy that's mean how well or how it's good the new treatment. Okay, so we just comparing between the two groups. There is control where we are giving them a placebo, and the other one is the case. And we know now how well this is new treatment by comparing to the person that they are taking the placebo. So demonstration of efficacy, and the other thing is measure absolute efficacy and safety of a new drug. So these we can say two reasons that are related to each other: demonstration of efficacy and measure the real efficacy and safety. Minimization of expectation, just again, it's a comparison between the case and the control, and if there is not a big difference, then we can realize that the new treatment it has not big deal with it and these things. And the other things is ethics. When we just give these patients some treatment that has no effect, like we didn't make any things abnormal or harmful to the cancer patients, and exactly no comparative information. So these are the goals of the placebo: demonstration of efficacy, measure the accurate efficacy and safety. It's also important as ethics to the cancer patient, no comparative information, and minimizations of the expectations. And then we go to the type of the intervention. As I said before, intervention, that's mean I do to the patient, I do something, I do effect to this, not observational intervention. That's mean I do something to the participants, and we subdivided it to the single arm design, and is a parallel design, and is a crossover design, and we have to know that all of these things, it's under the interventional. Actually, we discussed it before in the first lectures. We subdivided it into two types. We have interventional study and non-interventional study, and we said that the non-interventional study, it's observational study. Okay, now, okay, forget completely about the observation study and let's focus on the interventions. So the interventional studies design, we can subdivide it into three: single arm design, double arm design, or we can say parallel design, and we have a crossover design, and we wanted to know what is the difference between them. Single arm design, that we have just one group and all the members of this group receive the same drug. And why we do that? Actually, for dose finding, pharmacokinetic, post-marketing surveillance, indication of the therapy, actually uncontrolled, because there is no control group, just one group receive the same treatment, all the members receive the same treatment. And why we do that? Just to determine what is the dose or what is the maximum dose safety. We discuss also the pharmacokinetics. We look about the absorption, distribution, metabolism, and elimination. We discussed also the post-marketing surveillance, the indication of this drug therapy, and actually there is no controlled group. And that's why we call them as the single arm design. And we can now understand why it's intervention, because we give the treatment. We have to do something to these participants. We're just not observing things. And now we go to the parallel design. Actually, the parallel design is subdivided into treatment A and the treatment B. So we have two groups. Okay, so here it's about the late phase 2 and phase 3, because we know that the phase 2, it's a patient, and phase 3, it's about the efficacy and safety to make sure that is everything good. Robust study design, that means it's a strong and we can make sure that is everything okay, and simple execution of standard randomization mistakes possible. Yeah, that's maybe happen mistakes because we subdivided into two groups, and maybe there is some difference in these groups that actually they are not identical. And that's exactly what we mean exactly by the parallel design. Parallel design, they are parallel to each other. Group A receive a treatment, Group B receive another treatment, and that is important for the phase 2, phase 3. It's for the robots. It's a robust actual study design, and maybe there is a simple execution of the standards. Randomization errors, yeah, maybe happen. And the other thing is a crossover design, and we wanted to know what is a crossover design. So we have a group A, we have a group P. Group A receive a treatment, group P receive a treatment, and then we make a crossover between them. Pharmacokinetics, interaction studies, bioequivalence. Again, we study these things, but actually bioequivalence. Confounding covariates is reduced. In this, we are just exclude all the confounding factor, and we discussed before what is a confounding factors. It's like something that is related exposure, relate to the outcome, but it's not a sequence. It's not like a sequence or not a deal between the exposure and the outcome. And the statistically efficient recover fewer subjects. It's good for statistical. Doesn't mean that we have a lot of subjects. The issue of order effects and the issue of carryover between the treatment, like who will begin and who will end and these things. So these are what we call them as crossover design. So again, we have three types of the intervention. We have single arm design, double, or we can say parallel design, and we have a crossover design. So single arm, and from its name, it just one group receive one treatment, and that's an important for the pharmacokinetic, for the dose, for these things, post-marketing surveillance. And we have parallel design, that's mean we have two groups and each one receive treatment, different treatment, and that's an important in the late phase two, phase three. It's a robust um, study design, but randomization errors can may occur. Crossover design, we have a group A, group B, each one receive the treatment, and then we make a crossover, like we change the treatment. Because of that, maybe there is this issue of order, issue of carryover, and again, we study the bioequivalence, pharmacokinetics, and this stuff. Okay, then we go to something which is called pliding, or we can say it's a masking. So the blinding or the masking, what is the purpose of that is reduces the risk of the biases and confounding factors. Again, I explain what is a confounding factors. It's something that is related to exposure, but it's not as consequence of exposure and related to the outcome, but it's not like a factor between the exposure and the outcome. And the biases, we said again, is a systematic error. It's not a random error. And we discussed also the types of the biases. So blinding or masking, it's a purpose of that is reduce the risk of bias and the confounding factors. And we have four types of that. So we have open label, single blind, double blind, and double dummy. Um, there is a lecture that I discuss what is open label, and that's mean it's no one is a blind in each participant. I mean, the participant and the researchers know the experience and know everything about the experience, but single blind, it's one of them. They do not the exact information, so we call them is a single blind, and like it's one of them, maybe the participant, maybe the investigator. One of them do not know the information, and most of the cases the participant do not the information. Double blind, um, the investigator and the participant do not know the exact information, and there is a double dummy used to compare drugs with the different appearances. And we wanted to know what these things. I mean, we do the both, we give the same treatment. I mean, the active integrant of the drug, but the shape, I can say, of the tablet is different. That is exactly the double dummy or the double dummy. So, how to understand it is the double dummy technique. The patient always take a tablet and a capsule in a treatment A, as we see here. The tablet contains the active drug, and the capsule contains the placebo. In the treatment B, the capsule contains the active drug and the tablet contains the placebo. So again, this is exactly what is meant exactly by the double dummy, that the patient always takes a tablet and the capsule. He takes both things, take the tablet and the capsule. Okay, so this is the capsule and this is the tablet. Each group, I mean a group A and a group B. Okay, in a treatment A, he said that this tablet contains the active integrant. Here is the active integrant, but the capsule is a placebo or contains a placebo, so there is no effect of the capsule. And in the treatment P, the capsule contains the active integrant, but the tablet contains the placebo. And he didn't know, the participant or the participants don't know what exactly, where is the active integrant, where is the placebo, and that's what exactly we can say double dummy. So all these types of the masking, open label, single blind, double blind, and also we have something which is called double dummy. And now we discuss the end points. So the end points, uh, they are subdivided into many types: primary endpoints, secondary endpoints, exploratory endpoints, surrogate endpoints, composite endpoints, event outcome driven trials. So these are the endpoints. So the first thing is the primary endpoints. Central equation basis for the design and the sample size calculation addresses primary objectives of the trial, assessment of the main clinical benefits of a treatment. That's exactly what we see, the primary, and everything that we can say that the primary endpoints, that's mean it's the basic thing. So this is the central question basis for the design and the sample size calculation. So it like makes the basic questions, the basic thing, why we design this trial and where should we do this sample, and addresses the primary objectives of the trial, like what are the main things that should be done in this trial, and assessment of the main clinical benefits of a treatment, or what are the objectives of this treatment, and why we do this treatment, are the benefits of the treatment. And then is secondary endpoints, which is related to the primary endpoint, like helping endpoint, the primary endpoint. And then exploratory endpoint. And what I mean exactly by exploratory, when we just do these objectives, we can explore many things that we didn't plan for or we didn't expect that. And we have also surrogate endpoint. And the surrogate endpoint is the progression free survival against overall survival. Like we discussed the how it's progress and how it's uh, that happen. Composite endpoint combination of single endpoints, more precise, like we from its name composite, that we make it the combination of the single endpoints and we just collect the results and it's more precise. And there is something is called event or outcome driven trials, like what are the results of these trials, okay, or what is are the treatment or what are the results of these endpoints. So again, the types of the endpoints, we have primary endpoints, secondary endpoints, exploratory endpoints, surrogate endpoints, composite endpoints, event outcome driven trials. And it will be the next lecture. It will begin about the endpoints again and discuss the characters and everything about the endpoints. Thank you so much. I hope I understand you anything.